several databases with biochemical and genetic information, as well as bioinformatic tools. I will show you how to navigate some of these databases to learn more about specific genes and proteins. We will also take a closer look at how to analyze gene variants and their mutations. Let's start on the main Ncbi website. To get there, navigate to www.ncbi.nlm.nih.gov in your web browser, or just type Ncbi in your web browser and then click on the Ncbi website link. On the left side of the website, you will find a detailed list of resources that you have access to. On the right side, some of the most popular resources are listed, such as the BLAST tool, which I have a different video tutorial about. You can find the link to this tutorial in the description below. In the center of the page, you can find information on how to submit or download data. You can also find links to explore Ncbi's help documents and research projects. If you are interested in specific genes or proteins, you need to use the search box at the top of the page. To the left of the search box is a drop-down menu with a list of all the databases that are available for your search. Depending on what you are searching for, you want to choose gene, protein, or nucleotide from the list. Type in the name of a specific protein or gene and then click Search. We will search for the nucleotide sequence of the human CFTR gene, which is linked to a genetic disease called cystic fibrosis. The resulting list includes all the records that matched your search term in the selected database. For each item on the list, you can see its name, what organism it is from, the number of nucleotides, and its accession number. The accession number is a unique identifier given to a specific sequence in a database. On the left side of your search result page, you can filter your results based on species, molecular type, such as mRNA or genomic DNA, source database, and more. On the right side, you see the results by taxon. Another way to refine your search is by clicking on the advanced search link underneath the search box. In the search builder, you can narrow down your search, for example by entering a specific organism or looking for a specific search term. A simpler way to do the same thing is to go back out to the original search box. There you can add your specific search terms or organisms into the search box together with your protein or gene name. Note that some of the database entries are partial sequences. You want to select a full or complete sequence for your research. It might take a while to look through all the results until you find the right one. Once you have found it, click on it to get to the GenBank record for that sequence. The GenBank record has a wealth of information on your sequence, including its accession number, the names of the researchers who submitted the sequence, the date of submission, and much more. In the Features section, you can find information about genes and gene products, as well as regions of biological significance within the sequence, including their location. At the end of the page, you will see the actual nucleotide sequence. Most programs that analyze nucleotide or protein sequences require them to be in a certain format. The most common format is called FASTA. In this format, nucleotides or amino acids of a sequence are represented using single-letter codes. At the top of the page, click on the FASTA link below the sequence's accession number to view your sequence in FASTA format. You can now copy and paste the FASTA sequence if you need to transfer it into another bioinformatics tool such as BLAST. You can also download the FASTA file by clicking on Send to, Check File, and then click on the Create File button. Under Analyze the sequence on the right, you have the option to analyze the sequence using other bioinformatic tools. For example, clicking on Run BLAST brings you to the BLAST homepage, where you can compare the sequence to many other sequences in the database. You can watch Science Buddy's BLAST tutorial to find out more about how BLAST works. Further down the page, under Related Information, you can get more information on your nucleotide sequence.